Hi, my name is Crispr Wongpalee from the Department of Microbiology, Faculty of Medicine. My project is development of Crispr Cas12a based assay for rapid detection of Streptococcus suis. So as you may know, Streptococcus suis is a deadly bacteria. Infection can lead to hearing loss and sometimes death, as you can see across multiple news in Thailand every year. The problem here is that the current method of detection of Strep suis in the hospital usually take 2 to 3 days, which by the time will be too late for patients. So rapid identification of Streptococcus suis is highly needed for improved clinical outcome. So here comes the Crispr Cas. The Crispr Cas is a site-specific nuclease consisting of protein and RNA. The protein is a molecular sister, whereas the RNA function to guide the whole complex to DNA target through base pairing complementarity. Once the Crispr Cas finds the targets, it activates cleavage of the target. The Crispr RNA can be engineered into any sequences of interest, and this provides opportunity of using it for genome editing and also for, in our case, detection of pathogenic DNA. So the aim of this project is to develop Crispr-Cas complex to detect Strep suis with high specificity and speed. So how do we do that? We first have to search for DNA that are highly specific to suis. So by using bioinformatics, we retrieve about 94 DNA sequences of suis that are highly unique and highly conserved. And then we engineer those sequences into the Crispr-Cas complex. So here is the structure of the Crispr RNA. We selected a couple of candidates DNA and engineered them into the Crispr RNA in the blue region that you are seeing here. Then we also express the Cas protein in the lab, and then we purify them and assemble the RNA and protein together to form a functional Crispr-Cas complex that we can use for detection. So we did a couple experiments. We found that CRISPR-5 is highly active and specific. It can only detect Strep suis but not other pathogens. And the detection is at the lightning speed. It can detect as early as 3 minutes, and it reaches the maximum after 20 minutes. So in conclusion, we successfully engineered the Crispr Cas12a complex to detect suis genome with specificity and speed. So in the future direction, we are testing on clinical samples, and we are also adapting quick readout using paper dipstick. And my anticipation is that we can use this whole system in the hospital to detect Strep suis in a few hours after a patient arrives at the hospital, so we can advance the treatment 1 to 2 days earlier. And finally, I would like to thank the Murata Science Foundation for the support. Thank you.